I'm going to talk about trans-disease approach for therapeutic target predictions. Identifying therapeutic targets for diseases is important in drug discovery. However, the depletion of therapeutic targets is a major problem and has resulted in the recent stagnation of drug discovery. In this study, we propose a trans-disease approach considering disease similarities to predict new therapeutic targets using omics data. First, we constructed disease signatures and target signatures. For disease-specific transcriptome signatures, we collected the expression profiles of patients with various diseases and averaged these expression profiles. We constructed a disease signature for each of the 79 diseases, including Alzheimer's diseases and breast cancer. For target gene part of transcriptome signatures, we constructed two types of target gene part of signatures for target proteins. Target gene part of signatures represent gene expression patterns following gene knockdown or gene overexpression of target genes. We used 4,345 knockdown signatures and 4,040 overexpression signatures. Next, I will show trans-disease approach. Therapeutic targets are shared between pathologically similar diseases. So in this approach, we predicted therapeutic targets by multitask learning with disease similarities. First, we constructed disease similarity network based on cosine similarities between disease-specific transcriptome signatures. Next, we constructed multitask learning models. We use target gene part of the signatures as input data. We then prepared a predictive model for each disease. Each predictive model classifies input targets according to whether they are therapeutic targets for the disease or not. For trans-disease analysis, we learned predictive models jointly by sharing disease similarities. Here, we adapted a linear function that has weight vector W and explanatory variable X. We estimated weight vectors simultaneously by solving the following minimization problem. The first term is a logistic loss for all therapeutic targets. The second term is a ridge regularization, and the third term is a regularization term reflecting disease similarities. We designed the third term to bring the weight vectors close to each other if the diseases are similar. We evaluated the performance of our proposed approach. The left figure shows the result of inhibitory target prediction, and the right figure shows the result of activity target prediction. You can see that trans-disease approach performs the best for both inhibitory and activity target predictions. We predicted new therapeutic targets by using trans-disease approach. Here is a network which represents the distribution of therapeutic targets repositioned from the original disease classes to other disease classes. Each node represents disease classes. In the inhibitory target prediction, many inhibitory targets were repositioned between neoplasms and musculoskeletal diseases. This result suggests that trans-disease approach can predict novel uses of existing inhibitory targets for different disease classes. Next, this figure shows a small part of a newly predicted therapeutic target disease association network. For example, MUC1 is an inhibitory target of multiple myeloma and breast cancer. MUC1 was predicted to lead therapeutic effects in some diseases such as atopic dermatitis and inflammatory bowel diseases. We performed literature search and confirmed the validity of the therapeutic indications for four diseases with asterisks. Finally, we tried to make predictions for orphan proteins without known target disease associations. For example, we focused on STAMP-BP. STAMP-BP is an orphan protein and predicted as an inhibitory target for acute myeloid leukemia. We performed GO analysis by using STAMP-BP gene knockdown signature. GO terms of cell cycle regulation of G2M phase were enriched significantly. The regulation of this process is associated with suppression of cell proliferation. These results imply that our proposed approach can predict therapeutic indications for both existing proteins and orphan proteins without target disease associations. In conclusion, our proposed approach is expected to be useful for understanding the commonality among diseases and for therapeutic target identification in drug discovery.